Hello, everyone. I'm Dr. Carmen Pugliafito, and I'm here today with a good friend and colleague and outstanding vitreoretinal surgeon, clinician scientist, Dr. James Major of of Houston, Texas, where he is clinical associate professor at Houston Methodist Hospital and at Retina Associates of Texas. A big
Thanks,
group.
Yeah.
And he's done some interesting work looking at suprachoroidal injection. And we're going to really focus today on the basics of suprachoroidal therapy. This is something very new to most retina specialists. So what should we know about this procedure, this technique, this delivery approach?
Thanks, Dr. Pugliafito. It's great to be here. And it is an exciting approach because instead of being a new drug, it's a new space, really. So in that space, you can use old drugs, new drugs that we might develop, drugs that might be safer in that space. And so it's essentially the space between the cord and the retina, the suprachoroidal space, and it's a potential space, and it can be accessed in the clinic, similar to what you do with an intravitreal injection that we're so used to doing, but it's a 0.9 millimeter, a 900 micron or 0.9 of a millimeter in a flat tipped injector that's especially adapted micro injector. Once you get in, you sort of dimple the sclera, and once you get into that space, whatever medicine you're using in therapy then travels around the suprachoroidal space and gains access to the retina and the macula and the posterior pole.
So tell us a little bit more about the delivery system. What's
Yeah, it's a specialized micro injector that they've developed, and there's really two sizes to the needle. The normal size is the 900 micron needle, and that's basically the size of the sclera. You think about the sclera being about a millimeter thick. Well, this has just enough length to get through the sclera. And when you're injecting it, you actually have to dimple the eye in. And if you're in the sclera, you attempt to push the needle, you cannot, the drug obviously won't go through. And once you dimple the sclera and push in lightly, and we do it like you would a normal intravitreal injection, with iodine, povidone iodine prep, and a lid speculum, it then zippers around the suprachoroidal space. There's actually a separate needle number 2, which is a 1.1 millimeter needle. So you say, oh, this person has a thick sclera, I can't make it through. You can actually quickly change needles and use the 1.1, which is used fairly infrequently, but it's there. And once you hit the space, you inject it. So there is some technique involved in feel in terms of doing the injection. It's not just sort of a stick it in. You have to dimple it just enough to get into that space and use it. But otherwise, once you're used to it, it takes 30 seconds, a minute. It's quick.
How do you know that you're in the suprachoroidal space?
You can't inject if you're not. If you're not, and the typical, when you first start doing it, when you begin to inject, you think you're in the suprachoroidal space, and you cannot plunge the injector. You can't release the medicine. You dimple it ever so slightly, and then once it goes, there's no resistance. So it's all about feel and resistance on essentially the syringe to know you're in space.
Is there a chance of perforating the globe?
No, it's too small. The needle is flat tipped. It's not even a needle. It's really a micro injector needle. So it doesn't have a shape. It's flat and it's too small. So you're barely penetrating the sclera. So if anything, you edge on the side of not injecting or not being able to inject, where you're in the sclera, not able to push the medicine, and it's too short. You dimple it, and then it goes around.
I've heard that in some applications you use a thermal camera to watch the diffusion of the drug.
You can. They've radio labeled fluorescein, radio labeled the dye. Once you inject it in, instead of sitting like in a depot like you might imagine, well, once I inject it, it's just going to sit in a wad in that suprachoroidal space. It actually zippers around posteriorly and gains access to the macula and the posterior pole structures in the back. And the reason that happens is it's the same biophysics that really drives uveoscleral outflow. So the intraocular pressure is greater than the anterior suprachoroidal space pressure, which is greater than the posterior suprachoroidal space pressure. So thereby, when you inject it in the anterior suprachoroidal space, instead of just sitting there, it actually travels the pressure differential traveling from high pressure to low pressure. it goes from anteriorly and zippers around the posterior pole back toward the optic nerve. So the drug then sort of diffuses, and you can see this once it's radio labeled, and it's pretty easy using fluorescein light.
So what molecules can we inject?
Pretty much anything. You know, the size of the molecule, in typical like the Peachtree study and some of the uveitic studies, we use the old tried and true triamcinolone. But any molecule that's basically found in suspension, you can inject. So far, we've tried to inject in the eye. So there's, as some of the studies have talked about, wet macular degeneration, axitinib, potential molecules even for choroidal melanoma, because they gain access to the posterior pole. So any molecule that you can basically put in a liquid aqueous suspension, you can inject through, as long as it's not too viscous.
So where does the triamcinolone go, for instance?
So the triamcinolone, what's interesting about what's great about the triamcinolone is, you know, we're used to giving triamcinolone either in the subtenon triamcinolone or in the intravitreal space. But this triamcinolone, when you give it, say, in the Peachtree trial, which was the phase 3 trial, if you look at the actual numbers, it's found in once you give it suprachoroidally, it's found in very high concentrations in the retina and the macula, but very, very low concentrations, if at all, in the anterior chamber, in the lens, and the ciliary body, places that cause high IOP rises. So it has the wonderful benefit of giving you therapy where you need it in compartmentalization, but without the high IOP side effects. And that was borne out in the trial, too, in the Peachtree trial. People that received suprachoroidal injections of triamcinolone did not have corresponding IOP rises like you'd expect they would with a typical steroid injection.
Is there a depot effect?
There is, and there's an initial depot effect, and then there's durability in the drug too. And they found that drugs, as a generalization, drugs injected in the suprachoroidal space last longer than they would intravitreally. There seems to be the size of the molecule and the suspension that you use is cleared more, it's more difficult to clear by the choriocapillaris depending on the molecule size. But you can really have long-lasting durability, like 3-4 months with drugs that typically last 1 or 2 months in the intravitreal space.
What about gene therapy?
So this is exciting because, you know, we've seen problems. This gene therapy in the eye clinically works, but we've seen some very, very deleterious side effects like intraocular pressure that just falls to the ground, where the ciliary body becomes atrophy. There's iris body atrophy and atrophy to the iris. So gene therapy potentially, and then to me this is the most exciting area, but gene therapy potentially given in the suprachoroidal space, really you could get the efficacy that you need without the unwanted side effects of mounting an immune response as you do now when we injected the intravitreal space, and hence the the big negative of especially hypotony and lowering IOP with sort of destruction or detriment to the ciliary body. So this could potentially be a space where we could use this without the side effects and still have the wonderful efficacy that we saw.
Is there what tissues are transfected in suprachoroidal delivery?
So
the virus.
You mean when you give it?
Yes.
When you give it, it is absorbed mostly into the choriocapillaris, then diffused through the choriocapillaris and into the retina through the RPE and into the macula itself. So if you actually take and rabbit models look at the concentration of the drug that you injected anteriorly, you find the same concentrations posteriorly in the retina, retinal pigment epithelium, and choriocapillaris that you did anteriorly too. So it's not, the sclera being largely avascular is not involved there, but those other tissues it is. And what's nice is it's found in the same concentration in both anterior and posterior portions of the retina and RPE and choriocapillaris.
And some folks are using this to deliver a tyrosine kinase inhibitor for the treatment of wet AMD. Can you tell us about that?
Yeah, that's an exciting area. Anytime we get, we've long used just anti-VEGF monotherapy. And so you can combine any drug, this is one of them, like axitinib, and any drug that it potentially inhibits anti-VEGF, the different isoforms of VEGF, to use in the suprachoroidal space, and potentially add, added efficacy to both regular anti-VEGF or just a sole intravitreal injection with, say, a tyrosine kinase inhibitor or other
So
drugs.
how long does the tyrosine kinase inhibitor last in the suprachoroidal space? Do we have an idea about that?
Probably looking at 3 to 4 months, or potentially longer. So we don't know the exact answer to the question with this preclinical data, but definitely a prolonged effect on the order of many months versus, say, 1 month with your typical anti-VEGFs.
Why does it last so long?
It's a clearance. I don't know for certain, but it's a clearance issue in terms of the molecule not being able to be cleared from the choriocapillaris. And you think about, well, that doesn't exactly make sense, because the choriocapillaris is the most vascularized area of the body. It's densely vascularized. Well, it's not taken up well, and it diffuses well. Again, depending on the size of the pores and the openings and the permeability of the choriocapillaris. So that part is beyond my scope a little bit, but I do know that it has prolonged durability there.
So that's amazing. So really, it's a form of sustained drug delivery as well.
Absolutely. So, in big picture, you inject it in the front of the eye, in the anterior portion of the eye, it gets to the posterior pole because of differential pressure gradients. Number two, it diffuses throughout the posterior pole, getting to the retina and RPE and macula and areas where you want it to get. And number three, it minimizes anterior chamber penetration and its unwanted side effects that you could potentially get. And so, again, the sky's the limit for the suprachoroidal space, because pick a drug, whatever we have now, whatever comes in the future, it's exciting because it's not a new drug, but a new space to enter that has shown clinical efficacy in up to phase 1 and 2 and even phase 3 Peachtree trials.
Can you give us a step-by-step description of your technique in doing suprachoroidal injections? First of all, you're in the office, correct?
Correct. It is an office-based procedure. You do not need the OR. So it's very similar to an intravitreal injections, whereby the patient is prepped with povidone iodine. I use a sterile lid speculum and the typical sterile gloves and things that you might use for anti-VEGF injections. It's done. I typically do my anti-VEGF injections infratemporally, but this is done supratemporally for ease. It is also no subconjunctival lidocaine is used, because if you use subconjunctival lidocaine and give a subconj lidocaine injection, it distorts the conjunctiva enough where it's hard to see. You need to have a flat conjunctival surface and sort of a pristine white would-be sclera underneath. So you take the 900 micron needle, which is the typical needle we start with, and the microinjector, hold it perpendicular to the space. So we just use pledgets, like Q-tip, lidocaine pledgets to numb the area, and topical povidone iodine. You press slightly in on the eye, and then you attempt to depress. You depress a little further, and you actually dimple the sclera. It's when you see the dimple and you go in, then you really attempt to depress. If you cannot depress, you are not deep enough, you are in the sclera. If you go a little bit deeper and push dimple a little bit harder, sometimes a little more than you think you need to, then you should be able to easily depress. And what's interesting is, despite the topical lidocaine, the patients, something about medicine going into your suprachoroidal space, they feel it a little bit. So it doesn't feel like a pinch or an injection, but they usually will sort of comment, I felt that move around my eye. And so how we'd actually provide anesthesia for that, I don't know. It's not, painful. It's just sort of a unique experience, something zippering around your suprachoroidal space, but they feel it a little bit, not painful. And then you just withdraw and use I just move the conjunctiva over with a Q-tip and provide another drop of povidone iodine. If you cannot penetrate the sclera with your 900 micron needle and you've tried and you dimple in and you can't get through, then you would change to the 1.1 millimeter needle or 1100 micron and try the same effect. The patient's sclera is probably thicker and you just weren't able to penetrate it with your dimpling effect on the first injection.
Is the microinjector preloaded with the substance that you're going to
Yes,
inject?
the microinjector is preloaded with instructions. So whenever I've used it, it's always been part of a clinical trial. So it's preloaded with triamcinolone or axitinib or whatever drug you may be using. So it becomes preloaded. I would imagine in the future, if you were using this on a higher volume basis in the clinic, you would have to have a way to exchange, put in the drug that you needed to, or it would
It could be that
or it's come as a pre-filled syringe.
yeah, I think the way that the drugs are going to be distributed is linked to the delivery system. So the delivery system and the drug will come as a package, I
That
think.
makes sense. You open the package, put it down, and you say, "Here's your injection of, axitinib or whatever it is." Right? It's a pre-filled syringe. That makes sense.
Yeah. What's the volume of the syringe?
The volume, maybe a 5. You inject usually a 0.1 of whatever you're injecting. The needle is probably 0.5. It's about 5 times that long. It's a very skinny, typical sort of anti-VEGF needle, thin.
Well, this is exciting. We're at the very, very beginning of this. And I'd like to thank you for providing this introduction to your colleagues in the world of retina. So James, thank you.
Thank you, Dr. Puliafito. Exciting times for the suprachoroidal space, and I'm happy to be here. Thanks so much for having me.